Thank you very much, Martin, for the introduction, and would like to thank the World Cancer Research Fund for this opportunity to address this audience this afternoon. I particularly appreciated, actually, at the conference that for an organisation that's part of its remit, at least, is funding research, that there's a very nice atmosphere of exchange, actually, in cooperation with the research community to sort of try and identify the challenges and the way forward. And I think that's a really fruitful combination. Before I start the main body of my talk, just try to explain what my title is about to orientate you a little bit. And I just speak about one opportunity and one risk, I think, in this research area. What I'll speak about today is trying to demonstrate how the latest advances in understanding mechanisms of carcinogenesis, combined with some really powerful advances in technology, leave us with an opportunity to take a step change, to make a step change in understanding the role of nutrition, obesity, physical inactivity in cancer risk. And I still remember, you mentioned just before my PhD, I still remember my supervisor bursting into the lab, waving the latest issue of Nature, in which was reported the first evidence of a human oncogene, the RAS oncogene, in the early 1980s. And suddenly we had a target through which our environmental risk factors could act to drive the cancer process. And I think we now stand at a similar level of opportunity with respect to another category of changes, epigenetic changes. And these targets, which can be acted upon by factors in the environment, including nutritional factors and associated risk factors. I think that the risk, if you like, to match that opportunity is that we don't take this knowledge into the population research. And the reason behind my title, therefore, is that when we talk about translational cancer research, I think my feeling is that people mean to take the basic science into the clinic and to use this knowledge of mechanisms to design better treatments or biomarkers of early diagnosis or prognostic markers and so on. And that's certainly one value of that basic science. But I would like to argue during my talk today that equally, perhaps even more importantly, given some of the changing burdens of cancer, we can take that same knowledge of mechanisms and translate that knowledge into biomarkers, which can be applied in population-based studies to help us understand the causes and prevention of cancer. Hence the two-way translational research. So the structure of my talk is shown on this slide. I'll give a little bit of background about the importance of the environment, encompassing there, obviously, diet, in that, and the global cancer burden. Then I'll talk about the importance of being able to accurately measure exposure and the role that has to play in establishing etiology. Then I'll give some examples of what biomarkers may offer. And if you'll take the examples just as ways of illustrating potential rather than reading too much into the individual cases that I'll put forward. And then I'll finish with some priorities. So the agency for many years has been involved in estimating global cancer burden. And this was shown in a slide in an earlier talk today. But 2008 estimates of 12.7 million new cases of cancer worldwide. And as has been stated in the session on low-income countries, the majority of those already are found in less developed regions of the world. And I would encourage you to use the IARC GloboCan database online. You can discover a lot of information about cancer at a country, regional, global level, both now and projections into the future. And you'll see the major cancers listed on this slide in a global sense: lung, breast, colorectal, stomach, prostate cancer, and liver. One point just to bear in mind is that some of those cancers that are a little lower down the list in terms of incidence, notably stomach, liver, and esophagus, move up the list when you look at mortality. And that's because, of course, the treatments are much less effective for these cancers, but also these are the cancers that predominate in the developing world. And if you look at the projections into the future, I think for an international agency for research on cancer, it gives us a very strong steer as to why we should focus on cancer in low and middle income countries. These are the data for 2008, total cancer burden, and then split by more developed and less developed regions. But if you project forward to 2030, you see this gap, this inequality, if you like, really striking in the next 20 years. And so the burden of cancer falling very heavily onto the regions of the world that are least well equipped to deal with it. I should also point out that these projections assume no change in underlying incidence. So these are purely demographic driven, age and population growth. If you assume that there are some increases in risk due to the introduction of tobacco, for example, in these regions, the growth in obesity and physical inactivity, then of course these figures will be even more dramatic. And if you look at the cancer deaths, and again, perhaps just draw your attention to the situation in 2030, you see that that disparity is even more marked between the more developed and less developed regions. So to imagine trying to improve cancer services to treat the patients that will arrive with this increasing frequency, I think is not a strategy you could promote strongly, in the absence of at least a complementary attempt to identify risk factors and ensure that we do the maximum to promote prevention of cancer in these regions. So the basis of a lot of those projections and the increased number of cancers, as I've said, is due to the growth in the world population, but particularly in developing regions, and the aging of that population, again, seen particularly in developing regions of the world. So if we think about the major cancer risk factors, I've listed majority, I think, of the major identified risk factors on this slide. And I would like to draw attention to the importance of infections, although we won't dwell on that in this presentation. You know, more than 25% of cancers in developing countries have an infectious etiology. So if we're thinking of a comprehensive approach to non-communicable diseases, I think we mustn't leave behind the risk factors which are cancer-specific, such as infections, such as radiation, such as changes in reproductive patterns that influence cancer risk. So we cannot reduce the non-communicable disease response to a set of common risk factors. And I think this is an important point to take with us. So the agency recently completed a review of all Group 1 human carcinogens. This will be coming out sometime next year in a 6-part volume 100. And again, these will be accessible, and back volumes are accessible on the agency website. I pick up, though, over the last year and a half in this new job, quite a contrast in some ways. I mean, first of all, there's an emphasis, increasing emphasis on implementing what we know about cancer prevention in order to achieve that goal. And I put one example here, the European Partnership Action Against Cancer. In their documents, they cite that 1/3 of cancers are preventable. I think we could argue about the value there. But at the same time, we know that the majority of cancers have an environmental cause. So the potential, I think, for prevention is much higher. And I've labeled this here the etiology gap. And the reason to try to draw a little bit of attention to this is that I feel there is a danger of driving the cancer research agenda towards implementation of what's already known, to the potential deficit of detriment of continuing to look for risk factors, for what represents still a large proportion of cancers of uncertain etiology. And therefore, I think whilst we require research on how best to implement prevention strategies, and for those strategies we already have, we need to implement them into health services, I think at the same time for cancer research, it's important that we continue to focus on the causes of the disease. And particularly in many parts of the world, we have little information about the causes of some of the major cancers. So in terms of environmental exposure assessment, and I'm defining environment very broadly here to include nutrition, obesity, physical activity. I mean, we know many major common diseases have an environmental etiology. We recognize that currently exposure measurement is problematic. But also, I think we should recognize the emphasis that's being placed and the resources that are being placed into large prospective cohort studies that promise to elucidate the interaction between environmental and genetic risk factors for diseases such as cancer. And the one I know probably best is the UK Biobank, which I think in the first phase of recruitment of 500,000 people had a budget of something over £65 million. And I think that those studies, and there are others around the world, they're really predicated on the availability of accurate measures of environmental exposure. If we can't deliver that, then we will not be able to make best use of this investment in terms of understanding the causes of many chronic diseases. And I'd like to make the argument in the rest of my talk that, that biomarkers can contribute to several areas in response to this challenge. And to try and draw some attention to this, this requirement, I proposed an exposome to complement the genome. And the idea here was to have a set of tools which were equally powerful to assess environmental exposures as we have for the genetic components of human disease risk. And the symbol for this, I picked this male fiddler crab, mainly because through evolutionary pressure to attract a mate, the male fiddler crab has ended up with about 40 to 50% of its body weight in one claw, to such an extent that actually it can't feed itself anymore with this claw, so it relies on the small one to eat. But when I saw this, I just thought of the imbalance, really, between the effort and perhaps the funding to some degree, which has gone into refining the genetic part of the gene-environment interaction compared to the effort that's gone into really developing the tools we need for environmental exposure assessment. And it's really to draw attention to the need to get a better balance in those two areas if we're going to succeed in our goal. There are some real challenges, of course, in this. I mean, first of all, the scale and complexity of what might be described as the exposome. I mean, characterizing lifelong exposure to all the multiple risk factors that we might be interested in. We also would say that the exposome is far more dynamic than the genome, and it changes much more over time. And there may be critical periods in life when an exposure has a more important effect on risk than at other periods of life. But the message that I'd like to bring to you through some examples that I'll give in a moment is that even a partial characterization of the exposome could make major advances in our understanding of the causes of human disease, so that we shouldn't be daunted by the complexity of the challenge, in the same way that the genetic researchers were not daunted 15 years ago by the complexity of the human genome. I will put one caveat in just before I focus on biomarkers, and that is to say that these are one element of improving exposure assessment. So I recognize there are many other sophisticated approaches in terms of GIS, personal and environmental monitoring, increasingly sophisticated questionnaires that can complement and sometimes, of course, be better than biomarkers for exposure assessment. So what do they promise in population studies? Obviously, defining etiology by improving exposure assessment, reducing misclassification. I think identifying susceptible individuals within a population or subgroups. But also the contribution to the biological plausibility of exposure disease associations that we make in population-based studies. And I think increasingly, when the strength of association between an exposure and a disease is relatively modest, the evidence for the plausibility of that association at a biological level becomes increasingly valuable in making an overall assessment. I think these same markers can be useful not only in establishing the cause of disease, but making a first look at the impact of potential interventions or prevention strategies. So using the biomarkers as outcomes in intervention studies. And also increasingly, and I won't dwell on this here, but these markers and the data that they bring with them can be extremely useful in evaluating risks and in hazard assessment. So let me give you a first example, and this is not a nutritional factor, but a dietary contaminant, aflatoxins, which contaminate probably a quarter of the world's food supply. These are produced by fungi which grow on foods, particularly during storage in low and middle, well, in low-income countries. And they're toxins, natural toxins. Aflatoxin B1 is the most potent of these. This is a really strong liver carcinogen. And based on really a substantial amount of toxicology, how this compound is metabolized, where it binds to in the body, particularly the fact that this reactive epoxide is formed and binds to the N7 guanine position of DNA to induce a very characteristic G to T mutation. We've been able to understand something of the biology of this compound in the human body. But at the same time, we've been able to develop markers to measure that exposure in human blood samples. And in this case, we measure the aflatoxin bound covalently to albumin in peripheral blood. It's a very simple immunoassay-based biomarker. And what we've been able to do with that, it's really expanded our ability to study the relationship between aflatoxin and human disease. Before we had this biomarker, we were not really able to do good studies at the population level. We had to make estimates based on the amount of food people ate and the contamination by this toxin. And I just put some data up here by way of illustration. So these are 4 countries in West Africa, a number of subjects from different studies, and these are increasing levels of this toxin in the blood. So perhaps just look at the Gambia here, almost 1,000 subjects, and only 19 of them didn't have detectable carcinogen in the blood, aflatoxin in the blood. These were mostly young children aged between 3 and 8 years of age. And to get this amount of toxin in your blood, you really have to be consuming crops, typically in this population, peanuts, groundnuts, that are contaminated at levels well above those permitted for, let's say, dairy cattle in the European Union to protect the human population from transfer into the milk of this toxin. And you can see a similar pattern really across the countries. And just for comparison, I put some data from Europe and North America, where you see the vast majority of subjects have no detectable toxin in the blood. The important point from this slide is the characterization of exposure was completely transformed by this approach. What we had previously from the Gambia were data on the level of toxin in peanuts for export. And what was happening, of course, was the country was choosing the best quality peanuts to export because they would meet the stringent import controls in Europe for animal feed, not for human feed. Whereas what's being consumed here is subsistence farming level production of groundnuts and storage. This type of advance in the biomarker then allowed its application into epidemiological studies. And I just picked an example here. This is a prospective cohort study in China where baseline blood samples and urine samples were collected and then subjects followed, and a nested case control study of liver cancer conducted. And what you see here is that there is a risk of about 7-fold in individuals exposed to hepatitis B, strong risk factor for liver cancer, somewhat lower for aflatoxin, and then this combined exposure leading to this hugely increased risk, 60-fold in this case. But again, the general point I want to make is that it was the availability of the biomarker at the individual level that permitted this step forward in understanding the causes of liver cancer. And this was a key study in the IARC's classification of aflatoxin as a group 1 human carcinogen. Developing those sorts of markers, as many of you will appreciate, is not easy, and it's not easy to get funding to do that, because in order to develop that marker, we did studies originally where we measured over a 1-week period in a certain number of individuals in West Africa the level of toxin in their plate-ready, ready-to-eat foods, and correlated that with the biological samples collected repeatedly during that period. And in that way, we were sure that the thing we were measuring in the blood really represented the dietary intake of this toxin. And I think we really need to find a way to support studies such as that, which validate biomarkers before we apply them into large-scale epidemiological studies. And at the same time, we have to be thinking at a very early stage, working with epidemiologists to get the types of characteristics into the marker that allow application in large-scale studies. One of the examples where the technology is offering opportunities is in terms of then looking at the mutations induced by carcinogens in human samples. And again, aflatoxin was one of the early examples where there was a very characteristic G to T transversion mutation in up to 50% of the liver tumors that you find in regions where this exposure occurs. And that mutation was in one specific base in one particular codon of the p53 gene. And so the hope was that there would be a range of carcinogens that would be linked to certain induction of certain tumors by leaving behind a fingerprint, a pattern of their effects on the DNA of those tumors. But in actual fact, the number of carcinogens that have been associated with mutations in this way is relatively modest to date. But I think the opportunities offered by next-generation DNA sequencing are really striking. And in this case, I draw your attention to two sets of studies that came out just this year, earlier this year. I mean, one of them looked at a mutation spectrum of a lung tumor from a smoker, and a couple of studies looking at cell lines derived from human tumors. So what the investigators were doing here was sequencing the whole genome of the tumor and comparing it to the normal DNA from the same individual. So they're looking at every single somatic alteration in the tumor in relation to the exposure that had occurred. And let me just show you an example here. So this one on the right is a small cell lung cancer tumor. And these are all the mutations, base pair mutations occurring in that tumor. Nearly 23,000 individual events in that single tumor. What I've shown on the left here is the IARC database of the same tumor type, but these are 245 studies published on the p53 gene and the mutations found within that gene in that type of tumor. And I think just from a non-statistical perspective, you can see a very strong similarity between these patterns. What this means is from one single tumor, we really derive from a tumor that's strongly associated with tobacco smoking. You see an almost identical pattern to that generated from nearly 250 individual studies asking the same question in an individual gene. So I think this technology not only allows us to understand the genetics, the molecular genetics of a tumor, but it may offer us a fresh opportunity to read the information in that tumor in relation to exposure to certain risk factors. So is there an opportunity, for example, to look in things like esophageal adenocarcinoma in relation to obesity and people who are obese and not obese? Does that actually influence the type of mutations that you see? And does it give you some information about the pathways that are being modulated in that tumor development? So far, I've really focused on what I would say are a first generation of exposure biomarkers, which tended to focus on this classical mutagen carcinogen model of carcinogenesis. I think where the real excitement is also developing now is in relation to other mechanisms, particularly epigenetic changes. And I think here there's a really strong potential application to exposures such as nutrition, obesity, and physical activity. Maybe it's through some of these pathways that this group of exposures really influences disease risk. I'll give you a couple of examples just for purposes of illustration. The first one is a study from Senko Herseg in our institute at IARC. And what he and his team have developed is a very nice quantitative analysis of DNA methylation in minute amounts of DNA from body fluids. So in this study, they extracted DNA from very small amounts of plasma or serum. They amplified that DNA after modification to reveal methylation sites, and then did pyrosequencing to quantitate the level of methylation at different bases within selected genes. So again, the general point to make is that you can start to examine this type of mechanism in population-based studies with the type of samples that we find in our biobanks. The other one, which is looking extremely promising as well, is the microRNAs. These are stable molecules, again, in plasma and serum, and there are assays being developed to be able to measure these in human samples. And here's one example, again, I picked from the literature, a recent one on smoking-dependent changes in microRNA profiles. So these are normal lung specimens from a group of volunteers, either smokers or non-smokers. And basically, you just see a pattern of different either upregulated or downregulated microRNAs, generally correlating with smoking status in this study. So very preliminary, but the point is that in this case, the exposure is driving, or seems to be driving, and is at least associated with the pattern of these microRNA expression levels. Thinking about collaboration between epidemiologists and laboratory scientists, and it's interesting in that paper to see the study design. So there were 10 smokers, 7 males, 3 females, and 10 non-smokers, 3 males, 7 females. And the ethnic group were completely mixed. So you feel with a little bit more cooperation at the design level, the investigators could have drawn a lot more from this, this particular study. So the question is whether these technologies, looking globally at altered gene expression, can improve exposure assessment. I mean, do you pick up a pattern of change which relates to a given exposure? How do those alterations relate to dose? How stable are they over time? And how do confounding factors affect, or potential confounders, affect that association? I think, again, there's some promise here. And I won't go through this, but just draw your attention to a number of studies that have asked questions about exposed and unexposed groups to a variety of environmental agents and demonstrated differences in patterns of gene expression. So again, a lot of this research on transcriptomics is driven towards the clinic, but here's an opportunity to understand something about the mechanisms by which environmental risk factors modify gene expression. And I think that could be applied very nicely in the area of diet and cancer. Metabolomics is probably, I would feel, one of the most promising areas for the future of this field. And we've got really two overlying metabolomes, if you like. One is the endogenous one, profile that reflects an individual phenotype. And the other has sometimes been termed the xenometabolome. Now, these would be chemical constituents or metabolites which are signatures for particular exposures, either metabolites coming from the diet itself or maybe influencing the endogenous metabolome because of a given exposure. And I think the combined profiles of these have potentially exciting applications. And let me just dwell on that for a moment. So this is a study published quite recently. And the investigators here looked at 22 individuals and collected 40 urine samples per subject over 3 months. And then they asked the question, using a metabolomic approach, is there a similarity amongst the urine samples from a given individual? And what this slide is showing here is these are the 40 individual samples, each line, from each of the 22 subjects. And the message is that the inter-individual distances are far larger than the intra-individual distances in this study. And in fact, just 15 urine fingerprints per subject allowed you to predict which individual that sample would come from. And this is just another way of representing the data to show that individuals were really distinct in terms of their pattern of metabolites. At the same time, we have the possibility to identify chemical constituents of specific food groups, such as polyphenols in this case, composite chemicals, and also the metabolites coming from those chemicals in the food that are then found in the human body. And there's a very nice example of this, the work of Augustin Scalbert on phenols in foods, and identifying particular chemicals which can be a signature for a particular dietary factor. And of course, the possibility is that that same approach could be taken for different categories of foods, of micronutrients that we would be interested in studying. In this way, we can have a dual approach, I would say. In the first stage, one can use very controlled dietary studies, perhaps intervention, feeding studies, to link a metabolic fingerprint to a particular dietary factor or pattern. And then with those validated tools, move into the cohort studies and apply those same markers to look at the association between the level at, say, baseline in a cohort study and subsequent disease risk. There are problems with some of these technologies, and a lot of them come, I think, from the laboratory scientists applying directly their measurements to human samples without good interdisciplinary research teams. So one has to think, of course, about unmeasured confounding, bias through things like differences in sample processing, sample procedures to obtain the samples in the first place. And also many of these studies at the moment are small because of the high costs. Another related point that I want to make in terms of study designs is just illustrated on this slide. And I think this has been mentioned on the first day of the conference, that as with UK Biobank, many of the efforts we're making are going into adult cohort studies, large-scale prospective studies. But again, if the critical exposure is occurring early in life, either around the perinatal period or into adolescence, then these cohorts are not going to provide the answers to these critical questions. If I just go back to aflatoxin for a second, show you one slide here, again using our biomarker of exposure, where we looked at the impact of that toxin, which is present, as I say, almost ubiquitously in the diet of many developing countries. And here what we've plotted is the level of toxin against the height for age of those children in West Africa. And here we have children who are severely malnourished, and then children who are normal, let's say, height for age in this study. And you can see this very strong association between higher toxin exposure and impaired growth in these children. So here we have a very early life exposure. These children were being weaned onto solid family foods, and immediately their growth starts to be impaired. And we followed that up then in a longitudinal study where we looked over time at the child's growth trajectory and their aflatoxin levels. And again, just showing here in this table the lowest to the lower to upper quartile of aflatoxin exposure based on the biomarker, and this impairment of growth, almost a 6 centimeter growth over 8 months here, and just over 4 centimeters in the highest exposure group. But again, the general point here is that with the biomarker, we were able to conduct these studies of early life exposure, even transplacental exposure, that just weren't possible by questionnaire approaches. And there's another powerful example of this type of approach, this time using transcriptomics in a study in Thailand. And in this study, pregnant women were recruited in areas of high or low arsenic exposure. So this is another human carcinogen present in the environment. The women were characterized for their arsenic exposure using a biomarker. And then the investigators asked whether in the cord blood samples from children born to mothers with high or low exposure, do we see any difference in gene expression patterns? And what they showed is that there were very highly predictive patterns of gene expression correlated with prenatal arsenic exposure, and those clustered into certain biological pathways, which I won't dwell on here. But the point from this study is that this really early life exposure is already altering the biology of the child in such a way that could potentially predispose that child to a cancer risk later in life. So we can't afford to ignore the earliest stages of life when we're trying to elucidate causes of human cancer. So there may be critical windows during which exposure is important, but also we have to think of these exposures in a broader way and their impacts not only on cancer but on other disease endpoints if we're to evaluate fully the impact they have on human health. So I'm going to move now in my talk to two last areas. One, how these same markers can fill in some of the gaps about biological plausibility, and then very briefly an example of an intervention study. And in terms of biological plausibility, I'll give you two examples, one on demonstration of exposure and the other on a plausible mechanism. So the first one is a study of volatile organic carcinogens. So if you like, this is an occupational hazard related to diet and cancer. And there are observations that lung cancer in non-smoking Chinese women is higher, the risk is higher in those cooking in a Chinese-style wok fashion. And what the investigators did here was try to provide some biological data to support that epidemiological association. And they looked at a number of mercapturic acid metabolites in the urine from women who were cooking daily by these methods compared to a control group. And these are just 4 different carcinogens. And I'll perhaps focus here on benzene just as one example. You can see this higher level of exposure, and these are compounds which are present in the vapors from this mechanism of cooking. So I think the important point is the evidence that there is actually an increased exposure to some of these carcinogenic compounds in relation to that particular style of cooking. So that adds something to the epidemiological evidence. And the other one I'm sure is well known to many of you in this room. I think a very nice example, trying to understand some of the biology behind the association of red meat and colon cancer. And particularly the hypothesis that this may be due to endogenous nitrization. And in this study, and it's the type of study I think we could certainly see a lot more of in the future using some of these biomarkers, there's volunteers in a metabolic suite fed different diets, either high in red meat or high fiber diets in a crossover design. And the question was whether the level of nitrosa compounds in the feces or the associated DNA damage in colon DNA collected from the stool was modified by these different diets. I mean, again, going through this very briefly, but just to show here the data on an individual level of individuals consuming vegetarian versus high red meat diet. And this is the formation of the nitrosa compounds, the formation of the DNA adducts, and the correlation between the two markers. But here's an example where a hypothesis about mechanisms could now be addressed directly in human subjects rather than only in cell model or animal studies. And I think it's this type of translation of the basic science and technology into population studies that's so exciting at the moment. And finally, an example of where these same biomarkers could then be used to evaluate intervention studies. And one can think of different types of design. I'm just going to give you an example where it's a primary prevention in relation, again, to that dietary toxin, aflatoxin. So in this study, we were working, again, with farmers, subsistence farmers in Guinea and West Africa. We recruited subjects in 20 villages, 10 intervention and 10 control villages, 30 subjects per village. And we collected blood samples from these subjects at the time of harvest of the peanut, the groundnut crop, which is the major source of toxin in this population. And what normally happens is the farmers will then store that crop over a period of months, and the fungus proliferates and produces more toxin. So you see an increasing exposure with time. And all we asked was whether our biomarker demonstrated any differences by our intervention, which was quite a simple educational agricultural package. So typically farmers are storing peanuts either in these fiber structures here, but most commonly in discarded plastic rice bags. I mean, if you really want to produce fungus and toxin, this is one of the best ways to do it. You get high humidity, high temperature, lots of proliferation of the fungus. They also dry the peanuts on the ground. You might be able to see on this slide, you get a lot of humidity coming up from the earth. So we provided these mats for drying the peanuts on. We provided natural fiber jute bags instead of the rice bags. We provided wooden pallets to get the crop up off the floor of the storage facility. And we also provided some insecticide for the floor of the facility. A big problem is insects getting into the bags and generating further heat and spreading the fungus around. And we also worked with the farmers on selection of visibly moldy nuts prior to putting them into the bags for storage. So all these elements are used somewhat by the farmers in this population. We didn't introduce anything new. We just introduced it in a more systematic way, working with agricultural extension workers that already were in place in this region of Guinea. And to cut a very long story short, these are the data. So these are the levels of aflatoxin in the blood of subjects in the intervention villages in green and the control villages in red at the start of the study. And then you see this increasing exposure over time in the control villages and this flatter curve here with about a 50 to 60% reduction in exposure to aflatoxin in the villages from the intervention arm of the trial at the end of the study. And another way to look at that is to ask how many subjects didn't have detectable toxin in their blood at the end of the study. And again, in the control villages, it was about 2% and about 20% of subjects in the intervention villages. But again, the general point I'm trying to make here is that we can use these biomarkers of exposure as a first step to evaluate whether potential interventions have some merit before we scale up to very large studies. And I think this type of use of biomarkers is extremely valuable in this area of research. So my final point is coming back to the very beginning, this two-way translational research. I think we have this common soil of basic mechanisms, fantastic advances in understanding carcinogenesis. And there's a natural route of application for this into the clinic. There's, I think, a financial incentive for that. There's a great interest in developing better treatments of cancer. But I would like to strongly argue that if we can drag some of this same technology and knowledge into population studies, we can really make a difference in understanding the causes of cancer and developing interventions and prevention strategies. So, calling this, because we work in the Bilingual Institute in Lyon, a two-way translational cancer research. So my final slide, future challenges and future perspectives. I think it's beholden on us to support research into cancer causes and prevention in low and middle income countries because of this impending burden. I think we need to invest in exposure biomarkers, in particular, in a structured and coordinated way to complement the genetic analyses that we have available to us if we're going to understand the causes of common chronic disorders like cancer. We must therefore try and take some of the new methodologies and the new knowledge of mechanisms and bring those into our studies at a population level. I think there's a great gain to be had from prioritizing studies on biological plausibility to support the overall evaluation of exposure disease associations. And I think there's a particular need to think in terms of the life course of an individual and the exposure rather than only be driven by certain study designs and opportunities in adult cohorts. And I have no conflict of interest to declare. Thank you very much.
Thank you very much.